Welcome to Monohybrid Punnett squares. These are the basic Punnett square. This is the one you're going to see most likely on a test. And when you deal with monohybrid, again, we're talking with just one type of trait. And again, mono means one. But you have to know some key terms to do Punnett squares, and that's why we've been hitting vocabulary so hard. First of all, we need to know that if you're dominant, you're always going to be seen if you're there. If you're recessive, you're only seen if there's no dominant allele present. And dominant alleles will be expressed as a capital letter, and recessive alleles will be expressed as a lowercase of the same letter as the dominant. Don't change letters on this because you'll get confused when you go to the next section. We have homozygous. Homozygous means that the alleles are the same. Again, remember, two alleles make up a gene. And we will also sometimes call these purebreds. The opposite of homozygous is heterozygous. In heterozygous, the alleles are going to be different. And again, these are a lot of times referred to as a hybrid. And then our last two terms, we've got genotype. Again, when you look at the word genotype, I kind of see the word gene. That helps me remember that we're talking about the genetic information. So we're talking about the actual alleles. So if I ask for the genotype, you're going to be giving me letters. You're not going to be giving me descriptions. And then the counterpart to that is the phenotype. Again, remember this PH here reminds us of physical traits or photograph. So this is actually going to be a description of the alleles, of that particular gene. I have a 5-step process, and I guarantee if you follow those 5 steps in completion, no shortcuts, you can do Punnett squares. The 5 steps are this. First of all, determine the dominant and recessive alleles and name them. So like tall is usually dominant over short. So tall would be a capital T, short would be a lowercase t. Number 2, determine and record the genotypes of the parents. This is probably the hardest step of doing Punnett squares. But again, you're going to tell me what the alleles of the parents are. Again, you have to make sure to understand that vocabulary, and you have to read the question. Okay, step number 3, we're actually going to do the Punnett square. Step 4, we're going to determine the genotype of the offspring. So we're going to look at the boxes and see what the probability is that we would have a tall organism or a short organism if we're talking about height. And then the last thing, we're going to determine the phenotype, and that means we're going to actually describe what that offspring is going to look like. So let's do a couple of examples here. And again, these are in your packet. So you can follow along and fill in your packet as we go. So our first problem was cross 2 heterozygous tall pea plants. And tall is dominant to short. Well, first of all, we have to remember heterozygous means they're going to be different. We also know that tall is dominant. So again, we go with that 5-step process. Step number 1 was to determine the things that we're comparing. So we've got tall, and we've got short. And we know because we were told in the problem tall is dominant. So I'm going to say this is a capital T or uppercase T, and then my short's going to be a lowercase t. Step number 2 in our process was identify the genotype or the letters of the parent. In this particular case, they're heterozygous. Heterozygous we know means different. So my first parent's going to be big T, little t. My second parent is also going to be big T, little t, because they're both heterozygous. And at this point, we're ready to do our Punnett square. This is definitely a plug and chug process. So here's my square. I always like to put my first parent on the top just because I always do things the same way. That way I remember where I'm at. And again, remember, because the law of segregation, we're going to separate these alleles. So one T is going to go with this column, and the other T is going to go with this column. Same with my second parent. I'm going to put him on the side. And again, I put my big T here. I put my little t here. At this point, I'm going to carry everything down and everything across. So I've got a big T here, so I'm going to carry it down. I've got a little t here, I'm going to carry it down. I've got a big T here, I'm going to carry it across. I've got a little t here, I'm going to carry it across. And this is the possibility of my offspring. Now, one thing to be aware of. If I have big T, little t, and if I have little t, big T, it means the same thing, okay? So don't let that confuse you. Now again, when I'm doing my Punnett squares, I always like to put the uppercase letter first. But if you don't, and you come up with something with that, just count what you've got in each square. So, at this point, we're really just to do our last two steps and to determine what my offspring is going to be. And I'm going to have to do this over here. So step number 4 was determine the genotype of my offspring. So again, remember these boxes here, these represent my offspring. I've got a big T, big T. I've got a big T, little t. And I've got 2 little t's. So my big T, I've just got one of them. I'm going to make a little mark here so I know I've counted it. My big T, little t's, I've got one here, and I've got one here. So I've got a 2 out of 4. Actually, this is a 1 out of 4. And then I got my last box here, it's a little t, little t. So this is my probability for my little t, little t. And then the last step was what does these look like? So big T is going to be tall. And I've got one of them right here. My big T, little t, because I have a big T here, I have a dominant allele here, I know it's going to look like my dominant, so I know that I've got 2 more that I'm going to add here. My little t, I don't have a dominant allele, so I know that this one's going to be short. And I've just got 1 there. I'm going to add my 1 and 2 together, so I now have 3 out of 4. Or if you like to, you can say 75% and 25%. Just be aware, sometimes you'll see problems that say give the percentage or give the ratio. So if your problem says that, make sure you follow the directions. So that's my first one. That's my first example. Okay, let's try 2 more. In this case, I'm still dealing with peas. Yellow peas are dominant over green peas. We're going to cross a hybrid yellow with a pure green. So again, what are my traits that I'm looking at? I am looking at yellow, and I'm looking at green. I go back to my problem. It says yellows are dominant over green. So my yellow, I'm going to make a big Y. Now green would be a lowercase y, but be real careful and make sure that you know that it's a lowercase. And I like to put tails on everything. So there's my first step. Step number 2 says cross a hybrid yellow. Hybrid is different. It's a heterozygous. So I know I'm going to have a big Y and a little y. And I'm going to cross it with a pure green pea plant. Well, it doesn't say if this one's homozygous or heterozygous, but well, it does. It says it's pure, and we know that green is a little y, so we know parent number 2 is going to be 2 little y's. Okay, step 3 is do my Punnett square. So here's my big Y, little y for my first parent. Here's my little y, my little y. Again, I'm going to segregate them. I'm going to carry everything down. I'm going to carry everything across. And now I'm ready to see what my offspring are going to be. So, my genotypes, I look at the squares. I've got a big Y and a little y. I actually have 2 of them. I had little y, and I have 2 of those also. And then the last step was figure out what the phenotype is of these. So big Y, little y is going to look like my big Y, my dominant allele, which was yellow. And then my little y from down here, he's going to be green. And again, we have 2 out of 4, and we have 2 out of 4. Okay, that's number 2. Let's do one more. The monohybrid crosses, like I say, this is the one you really, really need to know because I will guarantee you this will be on the test. Okay, so I get tired of talking about P's sometimes, and so I get weird. So in this case, we're talking about teeth color. It says, in people, green teeth are dominant to purple teeth. Cross a heterozygous green-toothed person with a person who has purple teeth. So, again, what are my traits? I've got green, and I've got purple. Okay, it says green is dominant, so this is going to be my big G. And you can pick whatever letter you want, it doesn't matter. Purple, it's going to be my recessive, it's going to be a little g. Second step, what's the genotype or the letters for my parent? Okay, so heterozygous, heterozygous means different. So parent number 1 is going to be big G, little g. I'm going to cross that with a green-toothed person, with a purple-toothed person. Now, it doesn't tell us if it's homozygous or heterozygous, but it's purple. And we know that purple is recessive. So in order for purple to be seen, that means there can't be any big G's there. So this is going to be a little g, a little g. Do the Punnett square. Here's my first parent. Segregate those alleles. Here's my second parent. Carry everything down. Carry everything across. Okay, what's the genotype of my offspring? I look in the squares. I see that I have a big G, little g. I actually have 2 of those. And my other squares have little g, little g. And then 2 of 4 of them. What do these look like? Big G, little g. We got a dominant allele here, so we know that these are going to be green. Then again, there were two of those, 2 of the 4. My little g, again, there's no dominant allele here, so I know it's all going to be purple. And again, 2 out of 4. Okay, so that's monohybrid crosses. If you have questions on these worksheets, contact us, okay? We'll try to help you as much as we can. Okay, talk to you later. Bye.